What tyrosine does, tyrosine is made from another amino acid called phenylalanine. Tyrosine is one of 20 amino acids that are used to synthesize proteins. It's also essential in the production of some very important neurotransmitters, such as epinephrine, norepinephrine, and dopamine. This is tyrosine right here. And this shows how tyrosine is converted into the different neurotransmitters. This also shows how it's converted to the different neurotransmitters, but in more detail. You can see the stages it goes through to get to them. Tyrosine pathways. A ligand bonds to a tyrosine receptor, also known as RTK. A ligand then attaches with the 2-tyrosine kinase and becomes a transmembrane receptor and a dimer. Tyrosine bonds to ATP and takes of the phosphorylated dimer. Phosphotyrosine. It becomes phosphotyrosine because it takes a phosphorus from the ATP. Phosphotyrosine gets linked to the inactive relay proteins to connect with the phosphotyrosine and receptor. Once linked, proteins become active and create 2 cellular responses. The activated proteins indicate signal transduction pathways. Boom!
Yeah!
This is the signal transduction pathway. Tyrosine receptor domains. Receptor tyrosine kinase are transmembrane proteins with 3 main domains. An extracellular ligand binding domain.
Woo!
A single transmembrane domain. An intracellular tyrosine kinase domain. And that's all that's the domains right there. The extracellular ligand binding domain is responsible for recognizing and binding to specific signaling molecules that binds to the receptor. Initiates cell signaling pathways. Transmembrane domain. It plays an important role in anchoring the receptor to the cytoplasmic membrane and also by functionally connecting the extracellular to intracellular domains. Cytoplasmic tyrosine kinase domain. It functions as an enzyme that phosphorylates tyrosine residues on target proteins, transmitting signals from the cell surface into the cell to regulate cellular processes like growth and survival. This is the color-coded this is the color-coded pathway diagram right here. This up here is outside the cell, and these are the tyrosine receptor channels. That is the nucleus. And this first channel right here, it utilizes G protein. The proteins RAS, RAF, and MEK are part of a cascade that transfers cell signals from the cell surface to the nucleus, which leads to anti-apoptotic genes by activating ERK, which supports cell survival and proliferation. by increasing the expression of apoptotic genes. This pathway deals with phosphorylation and dephosphorylation. ATP is used by the protein kinases to phosphorylate amino acids by transferring a phosphate group from the ATP to amino acids. DAG activates PKC, which then phosphorylates other proteins. PKC can lead to anti-apoptosis by phosphorylating proteins involved in cell death, or by regulating the expression of anti-apoptotic genes. This pathway uses PIP2 and PIP3. Protein kinases these are protein kinases right here, lead to the proliferation of genes by activating intracellular signaling pathways. This leads to the transcription of genes that promote cell growth. Boom. Impacts of mutations. Mutations in tyrosine kinase can lead to the dysregulation of signaling pathways. This can potentially cause diseases like cancer. Mutations in the tyrosine kinase can also lead to drug resistance in cancer treatments. Cancer can be caused by the mutations making the tyrosine kinase constantly active, even in the absence of signal. Effects of pathway changes on cellular responses. Spocky. Phenotypic modifications. If mutations occur in the TH gene, which encodes the tyrosine hydroxylase, it can lead to impaired TH activity. This can lead to phenotypic modifications, which can cause disorders such as dopamine-responsive dystonia. Stop doing that. Infantile Parkinsonism and progressive infantile encephalopathy. Apoptosis. Apoptosis is cell death. It removes damaged or unnecessary cells. Changes to the tyrosine pathway can either inhibit or promote it. RTKs are cell surface receptors that can inhibit apoptosis by causing chain reactions in cells that promote cell life by activating the P13-AKT pathway. This pathway right here. When changes are made to the pathway, for example, inhibitors of RTK being introduced, apoptosis can increase. Increased apoptosis can be used to treat cancer cells. If they target a specific area, it can kill the cancer cells. But if apoptosis becomes excessive, it can lead to tissue damage. And that's the end. Final